What is allelic imbalance? Well, before discussing that, we have to remember that for any given gene, there's two copies that are inherited. These are homologous chromosomes, so they both code for the same gene, but one copy is the maternal copy, one copy is the paternal copy. Both of these copies are coding for the same gene, but they might be different versions, or they are different versions or alleles. So when we talk about these DNA sequences being transcribed into RNA, or the resulting products, and being expressed, we might have an imbalance in the expression. So normally you might expect 50% and 50%, so half of the expressed amount is maternal, half of the expressed amount is the paternal allele that's transcribed. So this is let's just say this is the RNA that's transcribed. But there can sometimes be an imbalance where you get a greater proportion of the transcribed or expressed genes being the maternal version, and then a smaller proportion or a different proportion is the paternal version, and vice versa. So you have an imbalance in the amount of one allele over the other for that given gene. So why does this happen? Why do we observe an allelic imbalance in some parts of the genome? So one reason that this can happen is because of changes to the genetic sequence or differences in the genetic sequence on parts of the DNA for that gene that regulate its expression. So for instance, the promoter region is known to be important for affecting transcription, right? That helps initiate the start of the transcription. But there can be cases where, let's say, the paternal promoter sequence is CGA and this one is CGT. And you can see there's a one-letter difference between the two different promoter sequences for the DNA. That's what we call a single nucleotide polymorphism, or SNP. So what an SNP is, or what this does, is that means that the promoter region is actually a different sequence for each allele. And this can have an effect because, remember, the promoter affects transcription. So let's say this single letter difference in the DNA sequence for the promoter, let's say it has an effect on how this gene is transcribed. And let's say it reduces the amount of paternal DNA that's transcribed. Then you could get a maternal, an allelic imbalance, where because of this difference in the promoter sequence, just due to one letter difference, you get more of the maternal gene expressed or transcribed compared to the paternal gene. Another example, or another way that this allelic imbalance can occur, is actually beyond the sequence. So when we talk about epigenetic changes, these are changes that affect DNA or genetic expression, but they don't change the sequence of the DNA. One example is methylation. So let's say the paternal gene is perhaps methylated at a certain point, and that epigenetic mark affects the transcription of that particular region of the DNA for that gene. That could result in an allelic imbalance. So the maternal gene is able to transcribe, but the paternal gene's expression or transcription is repressed by methylation. The sequence hasn't changed, but because of the methylation, the expression of that gene is inhibited. So those are two ways that allelic imbalance can occur.